Hello students, in the last lecture we have studied about the first step of thyroid hormone formation, iodine trapping. Today we will study the next step, synthesis and secretion of thyroglobulin. Thyroglobulin is a large glycoprotein which is secreted by the rough endoplasmic reticulum of epithelial cells of the follicles. When you will study about thyroglobulin, you will read that it contains 70 tyrosine residues. But thyroglobulin is not a monomer. It is a dimer. One monomer has 67 to 70 amino acid residues. So it forms a dimer before being secreted in the colloid. So one molecule of thyroglobulin contains about 140 tyrosine amino acid residues. So what we have to remember, that synthesis of thyroglobulin occurs inside the follicular cells, and then thyroglobulin is secreted by the vesicles found by the rough endoplasmic reticulum into the colloid, which is present in center of the follicles. Step 3 is the oxidation of iodides. See, in the last lecture we have studied that the sodium ion symporter at the base of follicle cell takes the iodide from the blood, and this iodide is transported directly into the colloid, and there is one more protein present here which is called as the pendrin protein, and this is iodide chlorine transporter. So this protein is pendrin. So the entry of iodide into the follicle cells is controlled by sodium ion symporter, and the exit of iodide from the follicular cell into the colloid is mediated by iodine chloride transporter, which is called as the pendrin. There is one very important enzyme present in the colloid called as the thyroid peroxidase. In some books you will read it as thyroperoxidase. In some books it's written as peroxidase. So thyroperoxidase is found in the colloid. It is not found in the follicular cells. So we will see how this thyroid peroxidase helps in formation of hormones. So the step 3 is oxidation of iodides. So now iodide have reached into the lumen, into the colloid, and thyroglobulin is also secreted into the colloid. So both are present. Thyroglobulin and iodide both are now present in the colloid. So iodine ion then move to the lumen of the follicle cell that border the colloid. Here the ions undergo oxidation. This is important, that the iodide ions will be oxidized. Their negatively charged electrons are removed. The oxidation of two iodide ions result in iodine. So now in the colloid, iodide ions are oxidized to form iodine, which passes through the follicle cells membrane into the colloid. So question we have to remember is iodine is oxidized to iodine by oxidized to iodine by it is oxidized by which enzyme? It is by enzyme thyroperoxidase. So the step 4 is organification of thyroglobulins. Iodinization of thyroglobulin is also called as the organification. The enzyme is thyroperoxidase TPO. Thyroperoxidase or thyroid peroxidase is using the hydrogen peroxide produced by dual oxidase protein. Now this thyroperoxidase is known to catalyze the following reaction in which hydrogen peroxide, two hydrogen ions and two iodide combine to form diiodine and water is released. Now thyroglobulin is already present here in the colloid. Thyroglobulin L tyrosine residue at the 3' end, 3' at the third position now reacts with hydrogen peroxide and iodide to form thyroglobulin 3-iodo L-tyrosine. So one monoiodination has occurred. This is called as monoiodotyrosine or modo 3-monoiodotyrosine. So mono, that's it is called as MIT, monoiodotyrosine. MIT in the next step, MIT at the 5 at the position number 5 is iodinated to form diiodotyrosine, which is 3,5-diiodotyrosine. So the second step is organification of thyroglobulin, which is occurring in the colloid. These tyrosine, they are still attached to thyroglobulin, and in the next step this MIT and DIT will couple to form the thyroid